Thank you very much, Tommaso, for the introduction and also for inviting me. So I'm going to talk about today something that we haven't finished, but I think I have enough. We have enough results to show you here, and hopefully this will be done very soon. So yes, my talk is organized in the following way. So first, I'm going to start with some motivation, which basically are certain biological experiments that serve as basis for what I'm going to present. Then I'm going to introduce the system of interacting particles. In this case, are going to be cells that describe the biological experiments and also how to go from this particle system to macroscopic PDE. And then this will serve as basis as well to introduce the inference framework for the parameter estimation that is the main part of the talk. Then towards the end, I'm going to talk about a bit future work and future avenues. Okay, so interacting particle systems are ubiquitous in nature, so we can find them in biology and life sciences, for instance, a flock of birds or school of fish. Also in social dynamics, so for instance, in pedestrian or opinion dynamics. And also in engineering applications, so for instance, drones and robots performing a certain or specific task, for instance, searching certain area. In all these cases, there are several ways of modeling these type of systems. So one of them is using stochastic or deterministic interacting particle systems. And in this case, these systems, they track each particle individually, and they are simple models because usually they are based on Newton's law. What happens is that in common applications, and especially in the ones that I just showed you, the number of particles is very large. So keeping track of each of one of these individuals might become very expensive. And also this system, as the number of particles increases, are analytically and computationally untractable. They are very high dimensional systems. So this is why in this talk, we are going to combine two different approaches, and we are also going to look at the limits when the number of particles n goes to infinity, and then we want to describe this system as a macroscopic density. Okay. Good. So let me start by introducing the cell-cell adhesion experiments, which are going to be our main motivation for the parameter estimation. So cell-cell adhesion has been studied for many years in biological contexts. So one of the first experiments was performed by Steinberg in the 60s, where at this point, he already observed certain segregation of the cell depending on the adhesion strengths. And also this is, cell-cell adhesion is a fundamental biological phenomena, which is related to tissue and organ formation, and it's a key ingredient in morphogenesis. Of course, since mathematicians were a bit slow, then we we got interested in this topic a few years later. So there has been some recent work from the mathematical side related to these cell-cell adhesion experiments. So what I'm going to show you here is a more recent experimental results from Togashi's group from Japan in 2015. And what we are going to observe is the same type of cell. So even though they are red and green, we have the same type of cell, but certain proteins at the surface of the cell have been modified so that we are going to see different strength in the cells, and therefore this will lead to different patterns in this experiments. So let's see. The video is playing, so this is good news. And let me stop it. Okay. Uh, okay. So as I mentioned, what we are going to see are depending on the cell strength, we are going to observe different patterns. So in the first case, what we have is a sharp interface between the two populations, the red and the green. In the second case, we have some interface, but also some mixing at the interface region. In the third case, what we see is that the red cells are pushing the green cells. And in the final case, we see pushing and some mixing between red and green cells. And what we want to do with this study is to, given some mathematical models that describe this particle system, what are the appropriate parameters in order to describe these four different cases. So this is our main motivation: how to estimate the parameters that give rise to these different cell-cell adhesion strengths. So, okay. Okay, so in the first part of the talk, I'm going to introduce the interacting particle system and its mean field limits. These are, I would say, somehow classical results. So I'm going to base this first part on these two papers and also on the model described in this paper from 2019. Okay, going back. So I'm going to start with a very simple toy model that will help me to introduce the actual particle model for the cell-cell dynamics. And this toy example is an aggregation equation, okay? So let's consider a system of n identical particles where xi is the position of the ith particle, and it's evolving according to this equation here. W, it's a radially symmetric interaction potential that is going to describe the pairwise interactions between particle I and particle J. Here, the scaling 1 over n, it's the mean field scaling, and basically it keeps the strength of the interactions of order 1. Okay, and this is going to be relevant. I haven't said anything about the form of this potential, so let's keep it as general as possible so far, okay? So in order to pass to the next level of description, we are going to introduce an empirical measure, mu n, that is basically a sum of delta functions centered at the particle's position. Okay, so I have a system of particles. I place the delta function on top of each particle, and then I sum over n. This using this empirical measure and using convolution with the delta function and properties of convolutions of measures, we can rewrite this equation here and have a gradient of W convolved with my empirical measure evaluated at the trajectories. Okay, of course, the methods that I'm describing here are not general, and you can obtain mean field limits of particle system using other tools, right? This is just one example of the whole plethora of techniques for mean field limits. Okay, so here what you have is this equation, right? But you still have n particles. So what you do next is to take the limit when n goes to infinity, and then you show that this empirical measure mu n converges in some sense to macroscopic density rho. And of course, here I'm hiding all the technicalities of the derivation, which are not important for this talk. Well, if for those that might be interested, if my potential is sufficiently regular, and if this empirical measure, mu n, at the initial time converges in some sense to the initial density rho 0 with respect to the usual Lebesgue measure in R2, then we know that mu n converges in the limit to the macroscopic density rho. Okay, so this is a bit hand-wavy, but this limit here we are going to consider that exists, and we can do it. So basically, when n goes to infinity, then we can write this equation here, where now we have rho. And this, when we look at this, we can identify the equation of the characteristics for this macroscopic aggregation equation, right? So this is the flux, which is exactly this. So basically, particles are transported. So this is the equation of the characteristics for this macroscopic aggregation equation. So basically, we started from the description of the particle system, and we ended up with the macroscopic mean field limit equation. Okay. This is a very general, and as I mentioned, is a toy example, but we can generalize this idea, for instance, for the case of two populations. So if I have red and green cells, as in the experiments, I can write the equations, sorry, I should be a time derivative here. I can write the equations for the two populations by defining the different interactions between the species. So W11, W12, W22, W21, which describe the different interactions between the species, but the idea is the same. So in this case, passing again to the limits, what we obtain is the system of coupled PDEs, where rho 1 is the density for the red cells, and rho 2 is the density of the green cells. Okay. Now we are going to keep adding ingredients to this equation so that we make it as closer as possible to the experimental results obtained. So another ingredient that we are going to add is local repulsion potentials that basically describe volume or size effects. So if some volume is occupied by some cell, then other cells cannot move to that place. We are going to do that. So we defined a new function, curly f, which now is going to be the sum of my attraction repulsion potential and this localized repulsion that is given by a delta function with some strength b. Okay, so if we substitute this f in our previous equation instead of w, we convolve with the delta function, and this is what we get. So we get the nonlinear diffusion term here. Okay, so at the PDE level, we have so far attraction repulsion forces and some nonlinear diffusion, where B1 and B2 are the nonlinear diffusion coefficients. This use of localized repulsion potential has been used before, and actually many years ago, from 1989, by a paper from a paper by Oschlager, where he introduced the same idea in the context of porous medium equation. This is a very nice paper, in my opinion. Okay, so the next ingredient is going to be some noise, because of course trajectories or cells don't move in a straight line motion. They have always some yiggling, so there's always some noise in the trajectories. So then we are going to introduce Brownian motions, so independent Brownian motion with some amplitude or strength given by sigma. So this is the new system of equations now, where remember that f, the curly f, was the potential W plus some delta function. And well, here we have the noise part. So at this point, we have a system of stochastic differential equations. So whatever I said at the beginning and this toy example that I presented at the beginning of the talk doesn't really apply to this case, right? Because the toy example was deterministic, and now we have some stochasticity in the equations. but the philosophy, it's more or less the same. Okay, so that's what we are going to see in a minute. So again, to pass to the limit, to the mean field limit in this stochastic system of equations, we define an empirical measure, mu n. In this case, this is going to be a random probability measure that again is going to contain all the information about the solution of the trajectories x1 to xn. And now, in order to pass to the mean field limit, we have to use some tools from stochastic analysis, namely Ito's lemma and Dubs inequality, so that we can bound certain terms. And then we can show that this stochastic behavior vanishes in the limit as n goes to infinity. And as a previous step to the final mean field limit PDE, we can write this system using the empirical measure in the same way that we did in the toy model case. So now we have the gradient of this function f convolved with mu evaluated at the different trajectories of the red and green cells. Okay, so finally, taking the limit as n goes to infinity, we get this system of coupled PDEs, where again we have attraction repulsion, nonlinear diffusion, and linear diffusion coming from the Brownian noise. Okay, and this is the final system that is going to serve. That is going to describe the experimental results that we showed you at the beginning. Okay. So the parameter inference part is based in this. Is based on this paper that, as I mentioned, hopefully we're going to finish at some point. And let me talk to you a bit about the data, the actual data that we have. So this is always the bottleneck in this type of problems. So in our case, we have selected trajectories. So basically, during the experiments, they pinpoint some cells, and then they tracked the trajectories of these cells over the time of the experiments. But of course, in order to apply the equations that I showed you at the beginning, so these stochastic equations, we need to know how this particle is interacting with all the surrounding particles, right? In order for the equations to make sense. And this is, of course, some information that we don't have. So how is this cell interacting with all the surrounding ones, and even with the other type of cell? This is something that is not available to us. Also, performing the inference on the SDEs is numerically very expensive. And what we would like is instead to do a combination. So we would like to use the mean field PDEs for the parameter estimation. Then we have a kind of a dichotomy, because our data are trajectories, and we want to perform the parameter estimation on the PDE. So here we have a bit of a problem. So how do we solve this? Let me remind you of the system that we have in mind. And at the trajectory level, we consider that this is our attraction repulsion potential, and this is the localized repulsion potential. And let me go step by step in this case. So here for the attraction repulsion, this is a very specific form, but of course you can think of other forms for this potential, as long as you reproduce the same different patterns that I showed you at the beginning. Okay, so this is only one case of interaction potential that works for the biological experiments. So here A describe the strength of the interaction. Here Mn are the different populations, red and green. Then we have one radius that is a shorted radius L, that describes the repulsion part of the kernel of the potential. And then we have another radius, capital R, here, that describes the attraction range of the potential. And of course, cells don't sense anything that is further away than R. Okay, so the potential is 0 for distances bigger than R. So this first term here describes the short range repulsion, and the second term describes the long range attraction. Then we have this localized repulsion form that well, describes somehow the volume filling, where B is again the nonlinear diffusion coefficient, and epsilon is a small parameter that, as epsilon goes to 0, this converges to the delta function that we wanted at the macroscopic level. Okay. So in short, what we want to estimate are this set of parameters theta, where we have A, L, and B. Of course, we have A for the two populations, so for the 4 different potentials. L, we have 2 different L's and 2 different B. Okay, so in total, we have 8 parameters to estimate in this case. Okay, so what is the inference framework that we would like to use? So to introduce the inference framework, let me go back to the one species example, where we have this, again, this SDE, where F has the potential, the attraction repulsion, and the localized repulsion, where all the parameters are in this function here, all the parameters theta. So our aim is, given a set of J trajectories, so xi star are my real trajectories, we want to estimate the parameters theta. In this case, the first problem arises, and is that X solves an SDE. So we cannot solve the inverse problem in the usual way, just minimizing the L2 distance between the real and the estimated trajectories, right? Because this, in this case, it will imply that I am looking at the derivative, integrating the square of the derivative of a Brownian motion. Okay, and Brownian motion has unbounded, is unbounded, so this this capital phi is almost surely infinite. Okay, so this is something that it's not computable at all. What we can do is to use the SDE instead, and then well, sorry, this F, it's I call all this term here F, capital F. Okay, what we can do instead is to use this SDE to rewrite this this term. And by doing that, what we have is this capital psi, which is the log likelihood function associated to the SDE, and is given by these two expressions here. Okay, so here what's roughly speaking, what we have done is to substitute here the SDE and expand this square. Okay, and then we get these two terms where this measure, sorry, this norm, it's a L2 norm, well, a Euclidean norm weighted by sigma, where sigma is a positive definite symmetric matrix. Remember that sigma is the amplitude of the Brownian noise. In our case, the trajectories are one-dimensional, so we have noise in only one dimension. So this this norm and this inner product here is only divided by the amplitude of the noise. Okay. Okay, so we have another problem, and is that in order to compute, or in order to minimize this capital psi, we need to know all the J trajectories, right? So we need to know all the trajectories of my trajectory I is interacting with, and this is again something that we don't have access to. So how can we overcome this problem is by using the mean field equations. Okay, so in the same way that I described it with the toy example at the beginning, we can use the macroscopic density rho. Okay, so we have now the gradient of F convolved with rho evaluated at the trajectories, at the real trajectories. Okay, so this is something that we can definitely compute given some trajectories xi star. But in fact, instead of minimizing directly this capital psi, this log likelihood function, we want to include some prior information about the parameters where theta star is the prior knowledge of the true values of the parameters theta, and C is the covariance matrix that can be tuned as well. So using Bayes and Girsanov theorem, we can show that in fact the posterior distribution of theta given some real trajectories xi star is proportional to e to the minus the error functional I, well, j. Okay, so at this point, we can do different things. So we either minimize the function psi, which will mean to compute the mean of the posterior. We can minimize j, which is computing the maximum approximate posterior estimator. Or what we actually do is sampling from the posterior distribution P, and this is our approach to use a full Bayesian inference to estimate these parameters. So we are not going to only find one specific parameter, but the distribution of the parameters. Okay, so for the two population case, this is the form of the functional of the log likelihood capital psi, where, well, it's analogous to what we saw for the one population case, but a bit more lengthy. Here, P1 are the trajectories for, well, the set of trajectories for the red cells, and P2 are the set of trajectories for the green cells. Okay? Good. So what do we do numerically? We use, as I mentioned, a Bayesian approach where we perform a Markov chain Monte Carlo method with a preconditioned Craig-Nicholson algorithm. Here, well, the advantages of using this is that, well, it provides posterior distribution of the parameters. Also, we have only one single parameter to tune, which is beta. This beta basically describes the step size during the search. So if beta is too small, this means that our MCMC is going to take very long to convert. And if beta is too large, then we are going to get a lot of negative output. So playing with beta can be tricky, but it's a parameter that can be tuned. Also, it allows for uncertainty quantification and has a simple interpretation of the results. The downside is that this methods, well, this algorithm requires many iterations to converge, especially when you are approximating 8 parameters, as is our case. Well, the numerical methods for the rest of the things, well, for the PDE, we use finite volume scheme, which is positive preserving, following some results from Jose in 2015. In the first part of our study, we use generated trajectories. So to generate these trajectories, we use an Euler-Maruyama algorithm. And to compute the convolutions, we use finite differences of the operators involved in the convolutions. To evaluate the real trajectories, it's just evaluated using a linear interpolation. So you remember when we evaluate this gradient of f convolved with rho, we evaluate them on the trajectory. So this is done by linear interpolation. Okay, so let's see some numerical results using generated trajectories. And in this case, we are going to consider only one population and nonlinear diffusion and Brownian noise. So basically, we are going to have a linear diffusion term, a nonlinear diffusion term, and attraction repulsion. Let's recall that the nonlinear diffusion is given by this expression here, where epsilon is a small parameter, and this is the potential for the attraction repulsion forces that I introduced at the beginning. So for this case, the true parameters are A equals to 1, L 1.8, B, which is the nonlinear coefficient 0.4, and R is fixed to 3. And here we have the result of 20,000 iterations of the MCMC algorithm. So on the left, what we have is a comparison between the true trajectories, which is the X, the blue bars that you can see here, with the trajectories generated using the empirical measure, which are the green bars. And we compare it to the macroscopic solution of the PD, which is the black line. So we have a very good comparison between the three of them. So the generated trajectories, the trajectories generated using the empirical measure, and the solution of the PD. On the right-hand side, what we have is the mean of the posterior distribution for the different parameters, and we see that it converges very quickly to the right values, A, L, and B. Okay, so for the one population case, also we explore a little bit more because we wanted to be sure that not only the results are good, but also that the MCMC is going in the right direction. So here we have A, L, and B on the left, and again A, L, and B at the bottom. So the main diagonal of this block, we have, the right value of A is 1, the true value of L is 1.8, and this one is 0.4. And on the off-diagonal squares, what we have is how the algorithm is converging to the actual value. So in all cases, we see that it's not doing anything crazy, and it's searching the parameter space appropriately. So this is a good sanity check in general. Then we move to the two population case, where in this case, first we have to tune the parameters in order to obtain the 4 different scenarios of the biological experiments. And here we have the 4 different ones. So we have the first case where we have a sharp interface, the second case where we have some mixing at the middle, the third case where we have one population pushing the other population, and the fourth case where we have pushing and a little bit of mixing. And again, we compared the trajectories generated using the empirical measure and the solution of the PDE. And we see that in the 4 cases, there is a relatively good comparison between the 3 of them. Okay, what we are going to do is we are going to do the parameter estimation only for the first case, so this case of the sharp interface. Here we consider that all A's are equal to 4, so A11, A22, A12, and A21, and the L's are equal to 0.5. And again, what we have on the y-axis is the mean posterior versus the number of iterations. And we see that these four first values, they converge eventually to 4, which is a true value. And the same for L, which remember that was the small radius of repulsion in the interaction potential. R here, I think, was fixed equal to 1 for this experiment. Okay, well, we did the same for the other cases, and well, the parameters are sometimes a bit difficult to tune, especially if the trajectories, for instance, are closer to the boundary and not close to the interface, then it might take a bit longer to convert and to obtain the right parameters in the other cases. Okay, so what about the real data? So this is the snapshot of the data that Togashi gave us, where we have trajectory 1, trajectory 2, and well, the data to reproduce these trajectories. But in fact, the truth is that this data is a bit of a mess. People, the ones that perform these experiments, they are no longer part of the lab, and understanding this data and translate them to the actual figures that I showed you before, it's not trivial. It's not trivial work. So far we haven't used the real trajectories yet, but we are trying to find a different approach to use real data. But this it will be very hard to clean all these trajectories that we got from the biological experiments. Okay, so as a future work, first we would like to finish the paper as a first step. That will be good. Then another possible avenue will be to inference of the interaction potentials following some previous results. So instead of estimating certain parameters at the interaction potential, which has a very specific form already, so can we infer the whole interaction potential, the whole function, instead of the parameters? This would be another possible direction. And instead of using the trajectories from the biological experiments, what we can also aim to do at some point is to use programmable robotic swarms as the source of data. So basically, we can dictate to the robots, or we can program to the robot the specific interactions that cells do, try to reproduce the experimental results, and then extract the data from the robotic trajectory. So in this way, we will have a set of data that we can definitely control, and we have a set of cells that we can also fully control in case we need different experiments or different type of data. Okay, so with this, I think I thank you all for your attention, and I'm happy to answer any questions.